Hello, friends and my dear students. Today we are going to see the another part of air microbiology in this online video class. We are continuously organizing the video class. So today we are going to study the control of microorganism by using various physical and chemical methods. So in this point we'll see how the physical agents and chemical agents they are used, how to control these agents by microorganisms, a number of methods, number of techniques, or number of physicals and chemical agents they are used to control the microorganisms. The first point under this control of microorganism or first physical agent, it is nothing but it is the radiations. A number of radiations they are used to control the air microorganisms. Its efficiency is dependent on the wavelength and intensity and duration, because each and every radiation has its own wavelength, it has its own intensity, and the time period is also fixed on that. On the period of that, the activity effectivity of the radiation depends. There are two types of radiations generally used: ionizing and non-ionizing radiations. In the ionizing radiation, there is the use of gamma rays and high energy electron beams. The sterilizing these ionizing radiations they are generally used for sterilizing pharmaceutical, medical, and dental supplies. It means where these materials are stored, within that these rays are used for in the industry's purpose for sterilizing the medical, dental, food preservative, and other industry process. If they are more penetrating, food is exposed to high level of radiations to kill insects, bacteria, and molds. It means that whatever the microorganisms they are present on the surfaces, outside or inside the food materials or any other parts, uh, due, uh, due to the exposures of such materials, the radiations are very effective. They kills the insects, bacteria, and molds, and the whatever the food material it becomes safe. Then non-ionizing radiations. In the non-ionizing radiations, generally UV, ultraviolet lights they are used. They are very penetrative. They are non-ionizing radiations. They generally use for damaging the DNA of the any microorganisms or any other pathogenic microorganisms. Once the DNA destroyed, means the microorganisms are killed. Their effective germicides wavelength, the wavelength of the ultraviolet light, which is generally in between 260 nanometer to 80 nanometer, in between that it is a very effective, it is lethal. It is lethal means it is effective against microorganisms. They are poor penetrations as compared to other ionizing radiations. UV radiations is only useful for disinfecting outer surfaces, because inside such materials, inside means inside the glass, inside the packaging materials, the non-ionizing radiation not penetrates. So but mostly the ultraviolet radiations they are used. Ultraviolet radiations are to 60 nanometer is quite lethal, but does not penetrate glass, dirt, films, water, and other substances very effectively. Because of the disadvantage, UV radiation is used as a sterilizing agent only a few specific situations. It means that for few specific situations these UV lights are used as compared to other sterilizing agents. Ultraviolet lamps are sometimes placed on the ceiling of rooms or in biological safety cabinet to sterilize the urine and any exposed surfaces. It means that inside the room, inside the hall, or inside the any, what happens whenever we the whatever the materials they are sterilized, whatever the surfaces of that materials are sterilized, but any other living humans they they cannot the UV is very effective against the humans or any living things, because they generally damage the DNA, and due to the damage of DNA there is the formation of thymine dimer. That's why it is very important to avoid ultraviolet light. Due to the avoidation we can avoid the whatever the light does not expose the eyes or any other parts, because due to which it is very effective. That's why whatever the switch button of UV lights, they are generally adjusted outside the door, so due to which what happens, the light not exposes on the body parts. And when we are inside the room, so we cannot use the ultraviolet light. Commercial UV units are available for water treatment. Pathogens and other microorganisms are developed when a thin layer of water is passed under the lamps. It means that nowadays what happens, the water filtration units in the houses or any outside also, what happened, ultraviolet radiations, they are adjusted, so due to which uh, they are important. Apart from the second most physical agent in the controlling the air microorganism is the HEPA filters. HEPA filters, it is very small size filters. They are generally adjusted in most of the instrument, or generally they are used in the laminar airflow. It is one of the instrument which are used in the laboratory as well as in the research level for carrying the aseptic, for maintaining the aseptic conditions. So for that HEPA filter, HEPA means high efficiency particulate air filters. They are used for filtration of small particles, captures of minimum of 99.97% of zero point 0.3 microns contaminants. It means that they avoids the entry of microorganisms within the laminar airflow. Means the filters, they are very small sizes. That's why these filters are important as compared to other sizes. You just see, this is the filter adjusted within the laminar airflow. Whenever the microorganism comes from one side, means whatever the air blowing from one side, so what happens, the microorganism, they settle on the surfaces of filters. They cannot enter towards the other side means in the laminar airflow. And filtration is a primary method of eliminating pathogens from the air supply. They generally use operating rooms, burn units, hospitals, surgical masks, pharmaceutical manufacturing facilities. So this is the laminar airflow. How the HEPA filter adjusts? This is the place, upper side. This is the place where the HEPA filters, they are adjusted. Uh, that's why the avoids the entry of microorganisms inside. Exhaust filters, exhaust HEPA filters, they are adjusted that upper side. The motor blower, due to which whatever the air blowing inside, the supply of HEPA filters, they are adjusted just between. Special light and electrical compartment is a special light and electrical compartment is adjusted on the the front sides. Safety glasses, view screens, and high velocity air barrier. It means that especially we are concentrating on HEPA filters where these adjusted, so due to which there we can avoid the entry of air microorganisms. Apart from that, what happens, any other physical agents are adjusted in the, they are used to control the air microorganisms, especially desiccations. Desiccation, it causes the cessation of metabolic activity followed by a decline of total viable populations. It means the use of desiccation methods, we can control air microorganism. The survival of microorganism varies depending on the following factors. It means there are different factors. On the basis of that, what we can avoid the microorganisms, the kind of microorganism, the material on which the organism is dried, the completeness of the drying process, the physical conditions to which the dried organisms are exposed, for example light, temperature and humidity. And gram negative cocci such as gonococci and meningococci are very sensitive to desiccation. It means that within a short period of times, these gram negative cocci, they are easily destroyed by this process. So this is the very important. So you just see the structure of desiccations. So this is the structure of desiccation, how we can keep the materials within the desiccator, and due to the desiccation process, it is one of the heating process, how the materials are dried. It means that whatever the moisture level, whatever the water percentage within the materials, they are destroyed due to the desiccation, and it means that the the material which is free from microorganisms. So this is the all these are the physical methods they are generally used to control the air microorganisms. So in the next lecture we will see the how the chemical agents, these are the physical agents, the how the chemical agents they are used to control the materials.